Now when your recombinant DNA is ready, your cloning steps are over, you need to transfer this recombinant DNA into a host cell. Normally the host cells are the bacterium, the E. coli. Different strains of E. coli are there, like the E. coli XL1 blue, E. coli DH5 alpha, E. coli GM109. Like this, there are several strains, and all these strains are ampicillin sensitive. As after the DNA transfer, they will become ampicillin resistant. And there are cases where you might have a different type of DNA, maybe a cassette of DNA, which has been designed for gene targeting purpose. So you might need to transfer that piece of DNA into some host cell. It may be bacterial host cell, or it may be a protozoa, or it may be an alga, or it may be plant cell or animal cell whatsoever. So there are several different methods of transferring these types of DNA, or you can call gene of interest, into the host cells. Let's see what are those methods. So there are 5 general methods of DNA transfer in bacterial cell as well as the other cells. The first one is this calcium chloride method or chemical transformation method. Then there is electroporation. Then there is lipotection, which is basically liposome mediated DNA transfer. Then microinjection, and there is a gene gun or particle bombardment system or Biolistic method for DNA transfer, particularly in plant cells. Now let us talk about one by one about each of these methods. The first method is calcium chloride or chemical transformation method. In this method, what is done is the Escherichia coli cells, which are normally not competent to take outside DNA, they are made competent by treatment with calcium chloride, and sometimes in addition to calcium chloride, some other like magnesium chloride or manganese chloride are also given. So due to this calcium chloride treatment, the cells become competent, and then they can be and followed by this heat shock. So chemical as well as the physical, a combination of these two methods makes the cells transiently porous, and they uptake the DNA from outside. When you add the ligation mix after the calcium chloride treatment, when the cells have become competent, and when you give a heat shock at about 42 degrees centigrade for 90 seconds or so, followed by immediate chilling, then what happens? The cell becomes transiently porous, and after that you have to revive the cell in yet another media which are a little bit nutritionally rich, so that these shocked cells get revived, and now you plate the cells in agar plates containing ampicillin, because the cells were earlier, they are not ampicillin resistant, they are ampicillin sensitive. Now when you plate them in ampicillin media, only those cells which get transformed are selected, the others are killed. So what is the mechanism? Regarding the mechanism of this calcium chloride method, it is yet not clear absolutely that how does the process occur. These transient pores are already present, or they are due to the result of this calcium chloride treatment. It is not exactly clear, but it has been found that there exists this kind of Beer's junctions that has been shown in the left bottom, that there are some junctions of outer and inner membrane. Those are called Beer's bridges or Beer's junctions, which are more in competent cells, and probably these are the pathways through which the DNA is taken in. And moreover, the calcium ion, which is positively charged, it coats the DNA as well as the negatively charged phosphoric acid residue of the lipid membrane, and which the DNA is also negatively charged, the phosphate of the membrane is also negatively. Because of that, both of them repel each other. So when the DNA becomes coated with calcium ion, the concentration is very important. If you are putting too much of concentration, then again the cell and the membrane both will become positive. The cell and the DNA both become positively charged. Again, the repulsion force will come into play. But the concentration has to be such that there are some negative charge still present on the DNA, and that interacts with the calcium ion on the membrane. and this is how the DNA gets attached to the or adsorbed to the membrane, cell membrane. Then when you this is followed by a heat shock. When you give a heat shock, then what happens? The cells were in the ice cold calcium chloride and the membrane was completely stabilized. So now you are giving a temperature imbalance, because of which suddenly there is a the destabilization of the lipid bilayer. It becomes a little bit fluidic and the DNA is taken in. The DNA also gets smaller in size because of the bivalent interaction with the bivalent. There is a static force which acts within the DNA, because of which the DNA becomes ball-like or supercoiled structure. Otherwise, it is a nicked circular DNA. So the size decreases, which facilitates it for the better uptake of the DNA into the cells. So this is how this calcium chloride method works. So this is electroporation. In this method, this is an electrical shock that is being given to the cells, and it has to be standardized at what particular voltage and capacitance you have to give the shock, so that the cells, all the cells do not die, but some cells they form pores through which the DNA or this electroporation is a versatile method. It also allows your RNA and proteins, whatever you put in the outer environment, outer milieu, that will be taken in. So usually a 450 capacitance and 150 volt is good enough. This is a picture of the cuvette that is used. The cuvette contains on both sides there are aluminum plates which connect with the electrodes within this channel that has been shown here, the plastic tray-like structure within which this cuvette fits in. You put your sample DNA within the cuvette, you put your DNA within the cuvette, and you set the capacitance and the voltage with this power pack, and then we give a pulse for milliseconds, certain milliseconds. Your DNA, your cells take the DNA inside. So this is very beautiful method. This is physical method of DNA transfer. After the shock, they must have to be revived, as it was also in case of the calcium chloride method. Then this is lipotection or liposome mediated DNA transfer. Liposomes are lipid vesicles. They are aqueous lipid bilayer vesicles. And if you make your liposome with positively charged phospholipids, they will interact with the DNA and they will acquire the DNA inside them. You can see how they have in the picture, they have internalized the DNA within them. And now since they are membrane bound structure, they will try to fuse with another membrane. With the mechanism of bilayer adhesion, bilayer fusion, the lipid vesicles, these liposomes, will get fused with the cells and they will deliver the DNA inside, and that DNA might get incorporated into the genome within the nucleus. So this is called lipotection because it is a method of transformation in eukaryotic system, particularly because here the cell wall, if there is a cell wall, it will be not feasible for the cell to fuse with the membrane. So it is possible with the protoplasts or with the animal cells. There is yet another mechanism by which the liposomes are internalized that has been shown in the left side of the picture, that the whole liposome gets internalized by an endosomal mechanism, and then the DNA, the inner liposomal phospholipid, they get dissolved and the DNA is expelled into the cytosol and they are taken in by the nucleus. So this is how lipotection, transformation in eukaryotic cell is called transfection. So transfection using liposome is called lipotection. So the next method is called microinjection. In this method, what is done, and the eukaryotic cell, it is usually done with the eukaryotic cell. The eukaryotic cell is kept fixed with the help of a suction pump, which is also a very narrow needle-like, pipette, micropipette, with the help of which you hold your cell, otherwise it will move. And with the help of a very fine needle, you can say it is a micro needle, with the help of which you can deliver your DNA. Sometimes the whole nuclei is taken out if you are trying to do some the embryo cloning or something like that, and you may implant your diploid nuclei within the cell, taking out the haploid nuclei, making your egg as a fertilized egg. So that is also done with this microinjection. But in eukaryotic system, particularly in case of Caenorhabditis elegans, this has been applied for RNAi research and many other different kinds of research. You can deliver your DNA, your RNA, or your protein whatsoever with this microinjection process. Then what happens when you inject your DNA? It goes and it recombines with the target DNA wherever it gets its homology within the target genome, and it incorporates itself. So this is mechanism which is used, method which is used in case of eukaryotic cells. And this one, the last one, is the gene gun, or you can call it particle bombardment or Biolistic method, in which you can see this is an instrument in which it's a gun-like structure. There is a barrel within which there is a bullet, this microprojectile bullet, and before the bullet, just in front of that, there are some microprojectile particles. Those are micro size, 0.6 micrometer size, gold particles or tungsten particles. Those are being coated with your gene of interest or DNA of interest. And when you charge this with a firing pin, what happens? This bullet goes and hits on the plates where it gets stopped, and through the vent, the particles, gold particles or the tungsten particles, they are thrown in forcibly within the cell, within the tissues that have been kept in the petri dish. Those are usually callus tissue of the plant cell. So most of these cells die because of this heating process. But there are some cells which acquire, which still remain alive, as well as they take the DNA within them. Sometimes when you want to transfer the DNA into the chloroplast or to the mitochondria, then also you can use this gene gun or bombardment system. And after that, the selection process is there, that you have to revive those cells which have taken the DNA as well as they are alive enough, and then you can go for antibiotic resistant screening process or so. So these are the methods of gene transfer in bacteria, plants and animals. I hope you like this video. Thank you very much.